Hi, thank you so much, and thank you for coming to hear all about the various things going on in TRIA, or that have gone on in TRIA. I'm going to attempt to give an overview of 10 years in 15 minutes. I don't know how successful I'll be. When I say you, or us, we, it means, like someone else said here, the royal we. And I've listed myself, Jorg and Janice here, but there's so many others as well in our labs and beyond, in our various collaborations and connections within the network too. So too many to list on a title slide. This is a timeline of TRIA as I see it anyhow. Around 2005 or so, Jorg Boehm, Colette Bruhl, and I put together a white paper that went to Genome BC that talked about a potential mountain pine beetle genome project. And it was, at the time, we thought it was a pretty ambitious thing. And it's become much more ambitious since that under the leadership of Janice and Jorg and others as well. It's been funded by Genome Alberta, Genome BC, Genome Canada for the first 5 years, and then NSERC from then on. Around the time we were putting together that white paper and the proposal itself, the outbreak peaked in central British Columbia, and a lot of the beetles seem to have spread over the Rockies. And in 2009, Kathy Cullingham's paper from 2011 mentions that around 2009, the beetles were first detected in jack pines. So our proposal was sort of right on the hitting the iron while it's proverbially hot. In 2011, the Grossmania clavigera genome was published. 2012, the mountain pine beetle transcriptome, and then in 2013, the pine beetle genome and the host pines, jack and lodgepole pine transcriptomes as well. And this is, I mean, many more arrows could be put into here. It would clutter up the slide. Since then, we've been doing a lot of work too on overwintering, on drought stress in pines and the defenses related to that. Very recently, Christine Chu has a poster nearby here, but she's published a nice paper on transverbinol accumulation. And if you look at Phil Batista's poster, we have some interesting stuff going on overwintering metabolite profiling too. Dendroctonus ponderosae is also called Dendroctonus monticola back in the day. They've been synonymized. 1902 is when Hopkins first named this insect. It was thought to be two separate species. They were put together. So I did a Google search using both terms, ranging from 1902, when you would expect to start seeing that in the literature, to 2008, when we first were funded. And there were about 4,000 or so published papers on either one or the other of these names. From 2009 until now, there have been 5,000 papers. So in the last 10 years, there have been more than as many papers as were published in the previous 100 years before that on this insect, which goes to show the interest that's taken off on this insect. And that's, and I mean, a small portion of those 5,000, but a small but substantial part are from this particular network. We have spent time looking at the mountain pine beetle genome. I mentioned in 2013 the genome itself, the first draft genome was published. And in that genome, we've seen many, many detoxification enzymes, including cytochrome P450s and glutathione S transferases. And one of the cool things there that Chris Keeling noticed when he was analyzing the genome was that there was at least one horizontal gene transfer event. That means that the beetle has in its genome a bacterial gene that's thought to likely help it with digesting sugars. And they also have a lot of wood-degrading enzymes as well. So this beetle, just looking at its enzyme, has evolved to face new challenges continually, and in some very interesting ways sometimes, by picking up a gene from another organism, for instance. And we can expect it to continue to do that into the future. We're currently improving on these data. In the back there, you'll find a poster with Chris and Victor and Felix Sperling on an improved genome for this insect. And these data from this project are all publicly available and are being used in some interesting ways. I have a Google Scholar alert that pops up when these things are cited, and some very interesting papers are coming out using the data from this project. We've also been characterizing mountain pine beetle gene products in Jörg Bowman's lab and in mine more recently as well. In Jörg's lab, there's been a lot of work on the pheromone biosynthesis of the insect and how the insect is able to produce these pheromones and what genes are involved specifically. And this is also, the genes, the proteins in the antennae that help it to search for new hosts or detect its own pheromone as well. And so we have some specific targets now for direct control. Interestingly, in that initial white paper, when I read through it the other day, we talked a lot about using drug discovery methods to go after ways of getting right at some of these enzymes in the insect and disrupting some of their biology that way. And we haven't got there yet, and delivery is probably one of the more difficult parts of that, but it's becoming more and more possible and feasible with the work that we've done. It might also be possible to manufacture some pheromone components using bacterial or fungal type systems. But there's many more targets to be found in this insect, many more interesting things about its biology. In my lab, Luke Spooner is working on various oxidative stress enzymes, for instance, and trying to figure out what they do. And any of these could be targets amenable to direct control measures down the line. In Colette Bruhl's lab, the G. clavigera genome, so the symbiotic fungus genome was published in around 2011. It turns out that this fungus, as we know, is integral to the insect, but some of the results from that genomic and transcriptomic work that she did, that the people in her lab did, showed how integral it happens to be. This area right in here is a portion of it shows the number of transcripts that are coming off of a particular portion of the genome, so genes that are being expressed or turned on when the fungus was in the presence of terpenoids, pine defense chemicals. And it turns out that in further work, they also found that the fungus itself can use some of the pine terpenoids as a carbon source for its own growth as well. So it's integrally connected to the insect in ways that we never even thought possible before that. Oftentimes I think we thought maybe it plugs up the resin ducts and other things, which it probably does, but it's actually helping the insect to detoxify the terpenoids in the tree. So it's very important to monitor the fungus as well as the insect as it spreads. We can't think of it as just an insect. We need to think of it as a disease system, and the strains of the fungus that the beetles carry with it as it moves into new areas is going to be important also. And again, these genome data are publicly available and are being used currently. The host pine transcriptomes of the lodgepole and jack pine were published in 2013 as well. Various highlights, and there's many from there that we could pull out, but one of them is that the terpene synthases, so the enzymes that are used to produce the secondary metabolites in the trees, are very similar to, or look very similar to each other, yet we also know from a lot of other work from this group and other groups as well, that in fact those trees have fairly different secondary metabolite components in them. So this probably relates to different gene expression, different genes being turned on at different times, or subtle differences in the activity of some of those enzymes. And there's a fair amount of work that needs to be done there as well, but we have a really good initial handle on it here and a good way forward on it. We know that this means that mountain pine beetles face a very different chemical environment in the area that they're moving into, and it also allows us then, using either chemical means or genetic means, to assess the populations of trees that it's moving into with some confidence to be able to say what the beetle is going to face. As we've heard already a bit, and we'll hear more today I'm sure too, we know that precipitation is a big deal now for host defenses, and so when you combine it with some of that transcriptomic data and the production of terpenoids, and then looking at, for instance, lesion length growth in jack pine and lodgepole pine under different drought conditions, we know that precipitation and defense genetics and the evolution thereof interact in a very complex fashion. And we also know that we need to then be taking precipitation and doubtless other environmental effects into consideration as we look at this insect move across its range. So there's more comparative work that can be done and is being done in these different in the different species of trees, plus the hybrids as well, under water stress and under situations of that sort in Janice's lab and elsewhere. In my lab, we've been looking at host colonization specifically. We found out that the beetles turn on and off a whole bunch of different genes, particularly turning on a bunch of different detoxification genes very early on as the adults enter into the trees. We also know now that the adult beetles are facing a ton of physiological stress, and they turn on all sorts of physiological stress enzymes as well, and we're trying to get a better handle on what some of those are doing in the insect themselves. And we also now know that the female beetle, when she leaves the tree she grew up in, isn't provisioning her eggs yet. She only starts provisioning her eggs once she actually lands on the tree that she's going to colonize and starts feeding. And at that point, vitellogenin, which is an egg provisioning protein, starts to build up in her body. And so she's holding off on reproductive effort until she's certain that she has put in enough effort to find a host tree. So this means that increasing the colonization effort through management tactics and strategies could reduce reproductive output, because the beetles are under a great deal of stress. They're holding off on reproductive provisioning up to a certain point until they get into the host tree. And Dr. Phil Batista in my lab, a postdoc in my lab, whose poster is over there, and Felix Sperling and I have some work going on right now looking at the evolution of these host colonization genes in the expanded range. We've also looked at detoxification or overwintering gene expression, and we found that the larvae are facing a very difficult environment. Not only do they have to detoxify all the leftover secondary metabolites that the parents have left after battling it out with the tree, but they have to do that while growing and getting a lot of antifreeze compounds into their bodies to be able to survive the upcoming winter. We know now that larvae use a very different set of genes in the autumn that they do in the spring. It's almost like they're two different animals to some extent, worried about detoxification, stress, and overwintering readiness in the autumn, and then switching over to development as fast as they can in the spring. And we have a very good understanding too of how glycerol is built up in these insects' bodies over the period of the fall. We've looked very carefully at the glycerol biosynthetic pathway. So late summer and autumn larvae are really susceptible to natural or management-induced stress, and you saw a little bit of that as well with what Kathy Bleicher just showed you. And we're investigating other antifreeze mechanisms. Trehalose is one possibility, and there's also a potential antifreeze protein that we have in our hands that we would like to look into a little bit more. And as also mentioned, Dr. Batista's poster over there, we're also investigating the overall overwintering metabolite profiles, and we see, I don't want to take his thunder too much, but there's a very large difference in how beetles from Vancouver Island act compared to beetles from Whitecourt, which is interesting in and of itself, and it means that there's probably different overwintering strategies and capabilities across the population. How much longer? 2 minutes, I've got it. Good to go. So we know that this insect is expanding. We've heard a lot about that today and in previous TRIA meetings, and you all talk about it too in your own profession as well. However, it turns out that this message isn't always getting out everywhere, and this is from the Center for Agriculture and Bioscience International. One of their pest sheets said that this insect has not yet appeared outside its geographic range, therefore it can only be considered as potentially invasive. So we haven't got the message out everywhere yet, but thankfully the message is being heard some distance even from us. And so this is near to Keith's cabin, I understand. This paper just came out about a week or so ago looking at the potential of mountain pine beetle susceptibility in the Great Lakes region. And the author writes, there are still many unknowns regarding mountain pine beetle in the Great Lakes region. However, as natural resource managers plan for the future, they may want to consider the potential arrival of mountain pine beetle in eastern pine forests when developing silvicultural prescriptions. So the message is being heard, and it's being thought of seriously several thousand kilometers from here, which is good. I think the most important part of TRIA, in reality, is the production of what NSERC likes to call highly qualified personnel, and what we call students and postdocs and others in our labs. TRIA-2, this is the numbers from TRIA-2, which is the last 5 years of TRIA-Net itself. About 83 HQP have been trained by this project, and these are the people that are going to help to get this message out further, and these are the people who are going to use the various tools either with this insect or with other insects or diseases in the forest. And this is arguably our largest accomplishment and the most important legacy of the past 10 years of research here, and none of this would have been possible without our many partners in the private and public sectors. So thank you very much, and if I have time for questions, throw them my way if Janice lets me.